So light enters the eye through the cornea and lens, which the lens is a little bubble above the, like, clear bubble that protects the iris and the pupil. And the lens is back here. And these two bend light and like focus it onto the retina, which is right here. And the retina contains photoreceptors, and these photoreceptors are rods, which I did in dark purple, which are more sensitive to dim light and motion, and then cones, which I did in blue, yellow, and red, because they detect color and like fine detail. And then photoreceptors convert the light into an electrical signal, and this electrical signal leaves the retina and eye through the optic nerves, which you see here. And they carry the information of both visual fields, which I color coded in red and blue. And then at the optic chiasm, which is right here, fibers from the nasal half of each eye cross to the opposite side. So the left visual field projects to the right hemisphere, and the right visual field projects to the left hemisphere. And I also color coded that, so I made after the chiasm, it's only red here and blue here instead of both on each. The fibers continue through the optic tracts, which are in orange, and synapse in the lateral geniculate nucleus of the thalamus, which I made these yellow little bulbs, and as you can see, like, they mostly gather here. And then for a better view of it, I also made a little sagittal view, so you can like kind of see where we are in the brain compared to like relative location of other things like the hypothalamus or the corpus callosum. And then I zoomed in on the lateral geniculate nucleus because it's actually very detailed. It has 6 layers total, 4 parvocellular and 2 magnocellular, and it goes down from 6 to 1. And then in between in blue is the coniocellular layers, and layers 1 through 3 in the green are obviously magnocellular, and they process motion in depth. And then layers 3 through 6 are parvocellular, and they handle color and fine detail, and which are kind of like the rods and cones, you know. And then the coniocellular layers between the colors process contrast. And then from the lateral geniculate nucleus, neurons project through the optic radiation, with the Meyer's loop and the temporal lobe carrying the upper visual field info, and the parietal pathway carrying the lower visual field info, to the primary visual cortex and the occipital lobe. So I did the optic radiation with obviously the same colors, blue and then red, but then I added some sparkle just to like tell a difference into the occipital lobe and primary visual cortex, which is kind of by the calcarean sulcus. And then here the brain begins a detailed analysis of edges, orientation, and visual patterns in a retinotopic map. And then from the primary visual cortex, signals are sent to higher order visual areas like V2 and V3 for like form and depth, V4 for color perception, and V5 for motion, before splitting into 2 main cortical streams. So I didn't do like label the different visual plays, but they would be in this area as well. But I did indeed label the cortical streams. So this is the dorsal stream because it's sticking up, and it goes to the parietal lobe, and it's the where-how pathway for motion and spatial awareness. And then this little red one is the ventral stream, which goes to the temporal lobe and is the what pathway for object and face recognition. And then together, this pathway transforms light into perception, allowing the brain to identify what we see and where it is in space.